This, in one slide, is why we're doing the course. This is a list of all the answers that candidates found hard. I basically drew a line in the sand and said, if candidates get it right less than 40% of the time, not what is the question, because I can't show you the questions, okay? I'm not allowed to show you the questions. If that's a disappointment, please don't get up and leave now. I'm not allowed to show you the questions that go in the exam, okay? When we wrote this course, we decided very clearly what was right and fair, and we said, we can't just show them, if you turn up and show them 400 questions, that will give you a slightly unfair advantage compared to everyone else in the exam. Now, you might want that unfair advantage, but we're straight backs. We're not going to do that. What we can show you is the answers that candidates found hard. And when we look at the answers that candidates found hard, this is what they get wrong. It's very interesting. I've got the easy one later in the slide. I've also got a slide which shows what the candidates found easy, and it comes up later in this talk. But this is what candidates found hard. So we can see embryology. The exact answer wasn't embryology, but it was I could see by the answer that it was embryology that they were asking about. Yeah, it was like 16-week gestation, 12, yeah, you could tell. Some words came up. Interestingly, false came up as hard and true came up as easy. I really don't understand that, but there were definitely some times where people guessed true even when the answer isn't true. Okay, so there's obviously a little bit of systematic bias there, that people are, if they don't know the answer or it's hard, they'll just guess the answer is false, when actually the answer, so they'll guess the answer is true, but when the answer is false. Interesting. There were all sorts of other words that came up, and it was so obvious how we wrote this course. So when you go through the lectures are planned on what you find hard as a group of individuals. So things that came up were words like, oh, foramina of Luschka and Magendie. Oh, welcome to neuroanatomy later. Okay, oh, ventilation perfusion mismatch. Adenovirus, Klebsiella pneumoniae. Oh, there's a few things here. I can see a pattern developing. And then here's some things that came up. Renal losses, potassium, fish, vitamin K, DNA analysis, microarray. These words are probably a mystery to you, I suspect. Karyotype, heparin contamination. There aren't that many words on this slide, and yet I could see straight away that there's a little gap in knowledge here. So, and the gap in knowledge that we're going to cover now is laboratory methods. Okay, laboratory methods. I'm going to split it down into 3 parts. I'm going to talk about genetics. We had that microarray fish stuff coming up, probably all a nice mystery. And then we're going to do some biochemistry. And I'm not going to try and tread on too much of the other speakers' toes. We've got the very, very wonderful Patrick Davies. We've got a competition. I've got to beat Patrick, okay? That can go on the videos, okay? Because I've got to beat Patrick. I'm determined. We were so neck and neck, weren't we? We've just working out which statistic we use to prove which one of us is better in the feedback. He's going to talk about renal. And then we've got some hematology in here, okay? We've also got some stuff coming on hemoglobin. My colleague Stephen Williams is coming to talk about hemoglobinopathies and hemoglobin physiology. So don't worry that we're not covering everything in this one lecture. We're going to talk, Jyoti's going to talk about the immune response. So we're going to do vitamin D and bone health. Bala's going to talk about that. And then renal anatomy and physiology. Now, I've handed round the room some post-it notes. What I'd like to do is you can split a pack between you, so you've got some. And what I would like you to do is, when you learn something that you think, oh my giddy aunt, that's really useful, I'd like you to write it down. And then at the end of each lecture, what I want you to do is to come up to the front and just stick your post-it notes on the learning board that we're going to be using. And that helps me to understand what it is that I've taught you, but it also will help you to remember what you've learned. In the wee small hours of the morning, or even better, in the wee small hours of the afternoon when you're doing the exam and your mind goes back and you can see your post-it note when you're in the exam, you'll go, I know the answer to this. Now, what was it? Okay, right. So, post-it notes are there, split them up. When you learn something, write it down, think, oh yeah, that's really good. So, genetics. There's some important genetics that you need to know. You need to know a bit about chromosome disorders, a little bit about single gene disorders, something about mitochondrial disorders, and imprinting disorders. At this point, there's usually someone in the audience who has a PhD in genetics. Anyone here got a PhD in genetics? There's nearly always someone. There's nearly always someone who's a proper. So I've got an MD in asthma genetics. So this is my area. It was my area, absolutely, in 2004 when I did it. And it's all changed since then. It's absolutely terrifying how much the technology has come on. So we were just doing wave sequencing when I started. Now, oh my goodness me, it's so much more complicated. And that has come into clinical practice. It absolutely is there in clinical practice every day. So this is a relevant bit of pediatrics for you. So I've got a question. Here's a question. Here's a good question because all the answers could be right. This is why it's a good single best answer question. And the question is, the following are types of chromosomal abnormality. Which of the following is most reliably detected by array CGH? Aha! Isn't that an evil question? This is exactly the sort of theory and science question we ask, and this is why you'll say these aren't clinically relevant questions, but it is. Because when you're faced with doing some genetic testing on a child in clinic, you need to know, should I do a karyotype, a FISH, an array CGH? Okay, you need to know what you need to ask for and why. Can anyone answer this question? I love it. Okay, you'll be able to answer the question after the talk. That's good. Okay, so let's just talk a bit about chromosomal disorders and some of the simple stuff. This is a slide that's taken from the Science of Pediatrics. This course is not meant to be an entire advertorial for buy the book. It is an advertorial for use the book. I will honestly say to you that when, as examiners, if we now see questions, when we're writing questions, the first port of call to write questions is to use the book, and the first port of call to check questions are right are to use the book. So if you're studying for this exam, I would suggest you use the book. This is a slide taken from it. There's an excellent chapter on genetics in there. What goes wrong with chromosomes? What goes wrong with chromosomes is you can have whole extra chromosomes or missing chromosomes. Examples. You might have anaphase lag that causes your sex chromosomes not to come out. I want to do a laser pointer, but I'm scared that... Oh, that's it. Okay. Oh, of course it doesn't work. It's a screen. I should know this by now. You can't use laser pointers on TVs. You can use them on the wall, and then they stop working. You see, I should have learned that by now. Yes, it's not a screen. I can't use my laser pointer. That's why it's not working. Okay, so I have to point with my hand then. Okay, so you can have anaphase lag. You don't quite get all the sex chromosomes coming out. You miss one. You get someone with Turner's syndrome, for instance. Or you can have non-disjunction as an error. That might lead to Down syndrome, for instance. That's the most common reason if you've got a whole extra or whole missing chromosome. You can have structural abnormalities. You can have a translocation. Say we take off, snap off this bit and put that there, and snap off this bit and put that. Oh, I've mixed them up. I've mixed up. That's now. This might be one of you because you've got all the right bits of DNA and they're probably pretty much in the right order. It might cause a problem because the breakpoint might occur in the middle or nearby a gene, and then that gene might not work so well. It might not. You can have an insertion. That's where we take a bit out and we get we add it. We've got an extra bit now. We've got an extra bit of DNA. Oh, that's going to cause problems potentially. Or we could have a deletion. That's taken a bit away. That's probably going to cause problems as well, isn't it? Or we could have an inversion. That's where we take bits and we actually swap them round. We can have a duplication where we add an extra. So in this case, it's not quite the same as an insertion where we've moved it into the wrong place, but we've got an extra one. So we've got gene A, say we've got the CFTR duplicated, we've got CFTR, and then we've got another CFTR next to it. We've got, oh, so we've got a duplication there. And then we've got ring chromosomes, where they're joined up at the end. All of these things can cause pathology. And indeed, when you look in the book, you go, this person looks like they might have a ring chromosome. But you have to know how you might look for that and what the test is to ask for, because it's not just one test anymore. When I was a lad, all you could ask for was a karyotype. So the right answer was always karyotype, wasn't it? Okay, and then they've invented all sorts of other tests. So now there's lots of tests we can do. Okay, so let's talk about what we can ask for. You can ask for a karyotype. There's some things you need to know about karyotype. There's some cool stuff about karyotype. I said that you hate, more than anything else, waiting for your results for the exam, don't you? How much more would you hate waiting for the results of a karyotype? Okay, that's important to understand, because chromosomes, coming from the Greek coloured body, okay, chromosomes are invisible, except in what situation? When do chromosomes appear? They appear in cell division cycles. So if you take people's blood and you want to look, you have to force the cells to start to divide to be able to see chromosomes, to be able to do a karyotype. So you have to put them into culture and get them to grow. And that's why it takes time to do. So it's not like CSI. You have to wait, you have to take it, you have to stabilise it, you then have to force it to grow before you can see it. Isn't that a cool bit of biology? That's a cool bit of biology. That's why it takes some time. And that's quite a useful bit of biology when you're trying to explain to parents, why is it my result's not back today when they're very stressed and on the ceiling? So it is something that I do use in clinical practice and say, well, we've got two ways of doing this test. We could just look at all the chromosomes under the microscope, but the reason that's not going to be back by 4 o'clock today is I send it to the lab, and it's not they're being lazy. They have to then persuade those cells to divide before they can then fix them and then look at the DNA. It doesn't, so that's a useful thing. It's comforting to parents. It makes them understand we're not just dragging our feet and being awkward. Arrays, CGH, we're going to go through all of these. So, karyotyping takes between 1 and 2 weeks to get back. That's important to know because the sample has to be cultured. And once the cultured cells are there, they have to be stained so you can look at them. And this is Giesmer staining, as I'm sure you all remember. And you can see extra bits or little bits. It's really, really obvious, actually, that the chromosomes, for those who don't know, one of my favourite museums in the world is in Valencia. And in Valencia, on the top floor, my kids are really bored of this museum. I've been lots and lots of times. So in Valencia, there's a museum, and they have an exhibit that's all chromosomes. And they've got like these big chromosomes, and then they've got all the genes on them, and they talk about what genes there are and how many gene numbers there are. It is so cool. It is so cool. One of the reasons that trisomy 21 is survivable and that the people with trisomy 21 are able to live is that there are relatively few genes. I think it's 590 genes on chromosome 21. So it's small, so there's less room, and it's got a low gene density, which is why it's survivable. And again, that's a useful thing to know. Whereas you've got a big long chromosome like chromosome 1 or chromosome 2, if you have a trisomy of chromosome 1 to chromosome 2, it's got many, many more genes on it. You're getting over 1,000. It's something like 2,200 for 2. It's quite a lot of genes, the one with most genes on. So they don't have equal numbers of genes. And they don't have equal gene density. There are about 30,000 genes that we know of, which is a small number, I think. If we start thinking about computers and just even what's in your PowerPoint and all the lines of code there, 30,000 is not a lot. And you think, yeah, we make these amazing complex individuals from those 30,000 genes. Karyotyping is really good for looking at big things. It needs to be 5 million bases long to see. I mean, we're kind of used to talking in billions now, aren't we? After the Brexit and financial crisis. And so, everything's just like, it's like a billion. Like a billion is in any way a meaningful number to a human being. It's like, it's just too big. But we're used to talking in these numbers. 3 billion base pairs in the human genome, okay? 3 billion. If you started counting out loud and say, you'd still be here in, I think it's 25 years' time, still trying to, so we need powerful information. So we can see big missing bits. And so if you are looking after a 14 or 15-year-old in clinic and you open their notes and they say genotype's normal, they've had a karyotype. Yes? Do you understand? And that's really important. You've got to understand that things have changed. So they will be missing, you will have whole bits of stuff that you've not really looked for. And the technology's moved on. It's going to move on again in your lifetime. And understanding that as a pediatrician is really important. So we can see ring chromosomes. They're really easy to see on karyotyping. So if you think you've got a ring chromosome, you're going to pick it up because it's obvious because it's a circle. And when you look at it on the you go, oh my Lord, that was a circle. That's not right. Okay, you can see rearrangements that are big. It's going to be a big rearrangement, more than 5 million base pairs. You can see inversions if they're big, because the staining's not right. But you then have to look quite carefully to say, you know, does the stain pattern, does the Gesmer pattern look the same on both of them? And remember, they're not lined up like this to start with, are they? They're like all over the place. For anyone who's not worked it out, 1 is the longest chromosome and 22's the shortest, apart from the Y chromosome. Okay. It's interesting, on this one, when you look at it, 21 looks like it's the shortest. It isn't about the gene number, though, it's about the length, because the gene density is different on each of them. There are 3 survivable trisomies, aren't there, to birth? Patau's and Edwards, 13, 18. So you're saying that really those are usually low gene density chromosomes. So you can actually understand, so now you know something new about genetics and science. Oh yeah, that's cool, isn't it? There's less genes. And the reason trisomy 21 is survivable is because there are so few genes there. Okay, fish. Fish was there when I was in medical school, which means it's incredibly old technique. Yeah, of course. Yeah, because they're accessible as a, but you could use anything, I suspect, yeah. It's accessibility, isn't it? You use what? Because blood's easier to take. Fish, a fishing trip. Yeah, very important. If you're going to do fish, you need to know what it is you're looking for. You need to be pretty certain you know that this child's got Williams. Yeah, that you have to know what you're looking for, otherwise you won't find out an answer. So you need to know the point of interest. But it gives really much more rapid results because it doesn't rely on that cell culture technique, yes? You can just take them and try and label them quickly. So you'll find out an answer much quicker. It's almost totally been supplanted by array CGH, which we're going to go through in a bit more detail because understanding how that works is useful. Array CGH. So by the way, if you're in the exam, unless the answer is obviously yes, this is a test we do for fish, the answer is no, fish is not the right answer, okay? It's nearly always the wrong answer in the 21st century. Okay, how does array CGH works? Basically, it involves colour, and it involves control DNA and the patient's DNA. And you use them to add to each other, and it's good for spotting differences. So you cut your DNA up into chunks, and then you match it. And if there's an excess of control DNA compared to your DNA, you've got a deletion. If there's an excess of your DNA compared to control DNA, you've got a duplication. Okay, so you've got extra DNA. Have you followed? Because there's a nice diagram. There's stuff in the book as well, but it's not very complex, but it's perhaps more complex than your brains can cope with at about 9:35 on a Saturday morning. It's useful to know, and it can spot smaller differences. It can spot differences down to 50,000 base pairs. So people are going back and doing array CGH on people who've had karyotypes done, and they're 13, 14, and they're not quite, you know, right, for want of a better word. They've got possibly something, I think they've got a syndrome, but I really can't see what's going on. I'm going back and doing array CGHs. Because it's there, and we've got single best answer questions which need 5 stems, It's probably worth knowing some of the other things that turn up in the lab. Quantitative fluorescent PCR. It really is, it's there to detect aneuploidy quickly. So in other words, an extra chromosome. And it's useful in prenatal testing because it doesn't need cell culture. Basically, it looks at these things called tandem repeats, which you see on all chromosomes, and it looks at those where you can get aneuploidies, which is 13, 18, 21, X chromosome, Y chromosomes, okay? And you look at the ratio of the product that you get, and you go, oh, wait a sec, we've got an extra chromosome 18 because we've not got the right, everyone else, we've got the right ratio for everything else, except 18's got an extra, whoa, wait a sec, we've got an extra chromosome 18 here. Okay, so this is useful. You've done an ultrasound scan, and you've got high risk, you want to know quickly, the mum wants to know quickly, you're getting close to points where it would be okay to do something to intervene. There's a rush on quantitative fluorescent PCR. As long as you know the only time it's useful probably as an answer is aneuploidy and quick detection, that's probably it. I don't think we're going to get into the details in the exam of tell me exactly how quantitative fluorescent PCR works, not least because I don't think I could talk you through it. But you will sit in clinic. Has anyone been in a genetics clinic? Yeah. Yeah.
If you had a chromosome 5 triplet,
yes.
would it have already
So
died?
not a triplet, but so you've got
Sorry,
three copies of chromosome. Yeah, so yeah.
So would it already have died at the point when this test?
Yeah, almost certainly. It's probably not survivable. So they're not looking for it because it's not survivable through to that stage of gestation. Yeah, and certainly not to birth.
So that's why you'd only look for those.
Yeah, exactly. These are ones where you were likely to have a pregnancy, but then you might have picked things up on the scan that are going, something's not quite right. Should we, we'll do a quantitative fluorescent PCR and we'll know quickly for you, Mrs. Jones. And this is the sort of situation where Mrs. Jones really, really does need to know quickly. Okay, has anyone gone to a genetics clinic? Or who's read genetics reports when they come back? Yeah? Do you recognise some of these things on the genetics reports? These baffling stages. I mean, they are horrendous. You sit in clinic, you get the result back and it says, Johnny is 46XYT2 semicolon Y P22 P15.1. Yeah? Anybody got any idea what that means? A little knowledge goes a long way, okay? I'll talk you through it. It's really quite good, because this is something where I've got a friend who's, any of you use Life in the Fast Lane? Life in the Fast Lane, the website? Mike Cadogan was at university with me, we're friends, we've been friends for a long time, drinking partners. I said, Mike, he's a world expert in snake envenomation. And I said, Mike, how are you a world expert in anything? Because, that's what you think about the people going to medical school with, basically. They can't be experts in anything. So and Mike's pretty cool. And he said, well, in the kingdom of the blind, the one-eyed man is king. Okay, so this is an absolute example of Mike's definition here. In the kingdom of the blind, the one-eyed man is king. You don't need to know all of these, but there are some useful things. If it's got an ish in, it means they've done a test using fish. A del is a deletion on chromosome 8. And it's on the P, it's on the short arm at position 23.1, and there's a termination at that point. It's dead easy. It's just the language. I mean, they could have, but of course you've got that result and you have to explain to parents in clinic what it means. Yeah, I love that face. That face is the right face. But that is what the parents want to know. Say, well, you've got this result, what does it mean? And that's all you've got. You've got this. It's like, it tells you exactly what it means. It's not difficult, but you have to have the confidence to look and read through it as some sort of code. So this is a 46XY. This is a boy. Okay. It's got a translocation, 2 to 5. And it swapped over at the P, the short arm, position 22 on chromosome 2 and position 15.1 on chromosome 5. Just as nomenclature, P, because the French describe the chromosomes, means the short arm, and Q is the long arm. They used Q because that was the next letter in the alphabet rather than P and L, okay? So if you were trying to work out what the French word for long is, which I spent ages trying to work, what's the French word for long that begins with Q? They just use the next letter in the alphabet. It's a joy, isn't it, science? It really is. This is fish, and this is saying you've got the right number. You've looked at 22Q11.2 here, and you've got 2 copies of this and 2 copies of that. That's normal. But it's telling you your test is normal. Whereas here you're missing a copy when you've looked, okay? So it's not as difficult as it looks. I feel like if I thought that was really, really clinically relevant, I might write some more questions for that in the theory and science exam. I'm going to wait to see how often it comes up in my clinical practice where I have to do it. But a little knowledge goes a long way in that. All right, end of genetics. You'll be pleased to know. Okay, biochemistry. I said I'd show you some things that candidates are good at, okay? So this is this slide. Here are some things that candidates are good at. Again, false comes up. They're good at that, because this is just where they're guessing. This is where more than 80% of candidates got it right. So if we looked at the answers that more than 80% of candidates got right, here's some things that you will find you know about. The optic chiasm. When it's anything in the brain, you all guess optic chiasm. And when it is the optic chiasm, you get the points. So you all guess it. I look at this. This slide just makes me giggle more than anything else. Oh, look, what else did they get right? Glucose, 6-phosphate dehydrogenase. So you know about that, don't you, children? Yes, you all do. 3 per second spike and wave. Hey, get in there. I'm so good, I'm getting some questions right in this exam. Okay, respiratory acidosis, metabolic acidosis, anti-tissue transglutaminase. I know what you know. To be honest, you know what you know. And some of you who are studying for the exam continue to relearn what you know, and you're good at that area, but you need to know the things you don't know. So what we're not going to do is we're not going to talk about acid-base balance. It's too easy. You actually know it. We're not going to waste time talking about celiac disease or thyroid function test interpretation. I know you know it. You do need to know it, and it's very important, that's right, but you know it as a group of individuals. So the way that you do better in the exam is know the stuff that the other people don't know and the bits that they avoid. And they are the dreaded anion gap, pseudohyponatremia. Oh, goodness me, it's so terrible. And urinary electrolytes. Isn't that the most horrible moment on your shift when they say, oh, can you just work out the fractional excretion for me? Okay, so we all know those areas are important. We all know that they're really, really hard. But this is where, if you get these questions right in the exam, this is how you get through. Okay. So let's talk a bit about anion gap. There's a description in theory and science about this and how to do it. There's also an error in the book, which I'm just so upset about, because I really, really do love the book. And we've managed to get a mathematical error in, which just goes to show that maths is difficult, even for the great Tom Lissau and the mediocre Dr. Carroll. Okay, maths is difficult. So we're good at identifying acid base. Okay, I'm not going to cover it. We're not so good at anion gap and interpreting its relevance. I don't know whether any of you actually even do anion gap in your clinical practice, but there are some tricks for the exam. To calculate an anion gap, you need to know the sodium and potassium, and you need to know the chloride and the bicarb. So if anion gap is likely to be useful, all of those pieces of information will have to be available in the STEM. So straight away you're going, why are they telling me the chloride and the bicarb? And you're thinking, well, if they're telling me the chloride and the bicarb, maybe it's because anion gap might be useful. Okay, so that's a useful tip. That kind of works for AKP as well. When you're getting more information than perhaps you would usually see in your usual username, you're thinking, ooh, maybe there's something important. Maybe there's a clue here that stops this question being ambiguous and actually makes it very clear-cut, but I'm not picking up on the clue. So the anion gap, let's keep it really simple, should be between 10 and 16. And it increases when you've got any unmeasured anion. And unmeasured anions would be things like lactate, organic acids, inorganic acids, and proteins. We've got a whole load of metabolic conditions where you get increased anions and you don't know what they are. This is the error. This is a question in theory and science, and I'm so upset about it. We've got enough information to calculate it. You've got a 6-day-old baby. They're breathing fast. They're not well. We're giving you lots of information. They've got a mild pancytopenia. They've got a chloride. It should say 98. It says 110. It's very annoying. A lactate of 8. So a bit of that's not great, is it? Ammonia of 60. And they're a little bit acidotic. pH is 7.29. CO2 is 2 though, and they've got no bicarb left. It's only 10. What's the most likely cause? And you've got a list of answers here. Urea cycle disorder. Why can we cross that off? Because the ammonia is only 60. So ammonia would be high in urea cycle disorder. Really important. That's definitely always one of those exam questions. Surfactant protein B deficiency. Not really. They're tachypneic, but they're not ventilated. And they're blowing down CO2, and their O2 is normal. Organic acid disorder. Could be. Let's go through this. Group B strep sepsis, common. If you've seen babies with group B strep, they're usually probably more acidotic, and they don't blow their CO2 down quite so effectively. Hypoxic ischemic encephalopathy, really. So we've got an organic acid disorder here. And the clue should be that there should be a marked raised anion gap. And really, if the chloride had been 98, there would have been, but we made the chloride too high and did our maths wrong. So when we added up, we said the anion gap's 31.6. It's not. The anion gap here is 12 less than that, 19.6. So it's raised, but it's not markedly raised. And I'd have made it markedly raised in the exam. And we threw it in the book, and we didn't spot it, and I'm so gutted, because I only spotted it. No one told me that was there. I spotted it when I came to write this lecture, and I went through and did the maths again and went, there's a maths error. The authors had sent it to us with the maths error in, and we hadn't spotted the maths error. This is one of the few questions I didn't write. Nearly all of the questions in the Science Pediatrics book I wrote. We had a few that were submitted by authors, and this was one of them. And therefore I hadn't checked the maths. So mea culpa. Right, some of the useful things to know, and that are really important clinical practice. Sodiums in diabetic ketoacidosis. They always appear lower than they are. Let me unpack that. What happens when you've got diabetic ketoacidosis and you've got very high glucose is the glucose adds osmolality. Don't worry, Patrick's coming back to osmolarity, okay? I know this is, and tonicity. This is a horrible area, isn't it? It's okay, we're coming back to it. What happens is the glucose pulls water out of the cells. It takes water out of the cells into the circulation, which falsely lowers the sodium. Have you followed that? It's really simple. You can all work it out from osmosis when we get you to talk through it, but when you're actually faced with it in clinical practice, remembering which way round it is really difficult. And there's a correction you do, which basically says if your glucose is very high and your sodium is normal, your sodium is not really normal. As you get your glucose going down, your sodium's going to go shooting up because the water's going to go back into the cells. There's a calculation. High glucose makes serum sodium appear falsely low, and you can calculate and correct going back. There are different calculators available online, and no one's really certain which one's right. Roughly speaking, if you've got a glucose of 50, my lord, then actually, if your sodium's being measured at 140, it's probably going to be somewhere between 153 and 159. The bottom line is, if you see someone who's got a sodium of 130 but a really high glucose, as their glucose comes down, their sodium's going to come back up again. Don't give them a load of extra sodium and start panicking that they're hyponatremic. That's an important clinical thing to know, because the glucose has pulled the water temporarily out of cells into the circulation, and it's going to go back as the glucose comes down. You get a similar effect with hyperlipidemia, and from a science point of view, that's important to know as well, that actually, lipids, lots of other things can have a tonicity that pulls water out of cells, which leads to a normal whole body sodium, but an apparent false hyponatremia. A useful and relevant piece of science. And it's all stuff you could have done from first principles when you did your GCSEs, when we talked about osmolality and osmosis. This is just the sort of thing we'd have, if we wanted to know, well, you're going to be good in medical school, can you think through scientific problems? We'd have got you to talk through it from first principles and work it out. And you'd have gone, yeah, I can work all that out. And it seems really obvious until you get in the exam and you're having a bad day, and then it seems really not obvious, okay, at that moment. There's only a few things you need to know for TAS in terms of urinary electrolytes. They are fractional excretion of sodium and the transtubular potassium gradient. How much do you hate being asked to work out the fractional excretion of sodium in clinical practice? It's in my top 10 list of jobs to delegate to the F1, F2, and not for me to have to do. And then, unfortunately, I end up doing it anyway because they just cry after about an hour of trying to do it. Is that right? Does that sound familiar to any of you? This is one of those jobs that is just the most. But it's a really simple concept, and it tells you where the kidney failure is going on. If it's pre-renal failure, your body will try and hold on to sodium. If it's renal failure, you'll lose sodium. That's why you want to know it. Just think about it. Let's just go back and do that again. Look, pre-renal failure, your body knows it's not got enough fluid. It's trying to hold on to volume, so it's trying to restore that volume. So what is it doing? It is, it's going to hold on to sodium. So you won't excrete much sodium. Renal damage, acute tubular necrosis, the kidneys had a hit, it's not going to be able to reabsorb, it's going to lose sodium, so you're going to lose a lot of sodium. So have you followed that? That's nice, simple science. Make sure you've got it in your head, because it makes sense, and once it makes sense and it's there, you won't forget it. Right. So what we're looking for is we want to know the quantity of sodium excreted divided by the quantity of sodium filtered. And to get that number, we do a calculation, which is urinary sodium times by creatinine, plasma creatinine, divided by plasma sodium times urinary creatinine. I know of no easy way of remembering which way around they are. You can work it out because it's telling you what's the quantity of sodium excreted divided by what's the quantity of sodium filtered. In clinical practice, you can look it up, but it is the sort of thing occasionally we want to really discriminate the guys who really know their science. That's the sort of thing we put that formula in and say, which of the following is the best formula to calculate? And then I could do plus signs or minus signs or multiply signs, and you're like going, I can't remember it. I know he told it us. I'm not promising it comes up in the exam, okay? This is important you hear, that this is just the sort of thing that if we were writing a question, we go, well, if we really want to know, do they really know the science? We asked a few formulas are useful to know in your life. The transtubular potassium gradient is also important because, and Patrick's going to come on to this in great detail tomorrow, and it's going to be a thing of beauty to behold because you're going to understand the kidney. You really are, in just 45 minutes. You're going to understand the kidney, and you're going to understand why this, the TTKG, is really important. That is the potassium in the urine times by the serum osmolality divided by the potassium in the blood times the urine osmolality, and you want to know how is the kidney handling potassium. How are the sodium-potassium exchange pumps working? And that changes. So, we know, I've covered, that the fractional excretion of sodium is low in pre-renal failure, high in renal failure, and it's increased by diuretics as well, because they're telling you to lose sodium. You know that, Luke, you're pretty good at the diuretic bit. Transtubular potassium gradient is usually more than 5, but it's less than 3 if you haven't got any aldosterone. There are lots of those nasty little science questions, aren't there, about aldosterone? And it's more than 10 if you've got too much aldosterone. So it's a really clear-cut answer. It's telling you what the kidney's doing and how it's responding to aldosterone. Cool. Oh my giddy aunt. Coagulation. Isn't it also confusing? It is really confusing. It's really hard. Extrinsic pathways. On the train yesterday, I was looking at this and I was just going, oh God, I really still hate the common pathway, extrinsic pathway and intrinsic pathway. And I should have known it since about year 2 of medical school. And every time I come to it, it's like, this is all new. This is all new. I'm sure I've not seen this before. I'm going to tell you a story. Actually, I'll tell you this story first. I won't answer the question first. I thought of a great question. I'll tell you what the great question is. Child's got ITP. All right? You've all seen someone with ITP, yeah? Just write down a piece of paper. PT, low, normal, high. APTT, low, normal, high. Great question. Do you know the answer? You look confident? I'm going to tell you, it's all normal. The coagulation's all normal. Okay? It's great fun when the F1 stroke F2 stroke ST1 stroke ST4 says, yeah, we're just seeing child, we think we've got ITP, we're just waiting for the clotting to come back. It's like, it's going to be normal, okay? It's a platelet problem, okay? So remember, platelets do not affect. And that's a good and fair theory and science question, isn't it? Yeah? That is it, because it's a common problem. You need to understand platelets are not going to affect your APT and PTT. They're going to be affected by the intrinsic and extrinsic pathways. So here we go. We've got a 4-year-old boy, presents with a nosebleed. You can see a lot of those over the years. He's got an APTT of 67 seconds and a PTT of 12 seconds. What's the most likely diagnosis? Well, we've crossed off ITP already, haven't we? Yeah? So that's good. We're going to go through it. Okay, so normal hemostasis. We've got platelets, clotting factors, and the vessel wall. That's Virchow's triad. Helps you to clot. We've got an extrinsic pathway and an extrinsic pathway. We've got the common pathway underneath it. Okay? If you've got problems with the extrinsic pathway, it's going to be detected by the prothrombin time. And if you've got problems with the intrinsic pathway, it's going to be detected by the activated partial thromboplastin time, APTT. If you've got problems with the common pathway, it will affect both. So now you just need to work out which bits affect which, and then you'll be able to go backwards and work out which bits are missing. So, if you've got not what this patient's got, a high prothrombin time but a normal APTT, you've got an extrinsic pathway problem. So either you're on warfarin, like that's going to be the answer, isn't it? You're on warfarin. Or you've got a factor 7 deficiency. That's not likely to be the answer, but that's pediatrics for you. We do see some rare stuff every now and again, okay? High APTT, normal PT, that's our patient here. It's an intrinsic pathway problem. It's going to be either haemophilia A or B, factor 11 or factor 12 deficiency. I seem to think Christmas disease is coming to mind. Von Willebrand's disease, lupus anticoagulant, or heparin. If you've got high PT and high APTT, it's usually disseminated intravascular coagulopathy, in my experience, DIC, but it can be fibrinogen or vitamin K deficiency. So you give these kids vitamin K as stabilization. Okay, let's go back to our case. What's he got? He's got a high APTT, but a normal PT. So, what's the problem going to be? Digital trauma. He's sticking his finger up his nose, giving himself nosebleeds. That's the most common cause of nosebleeds. Is that the case in this child? No, because his clotting would be normal. Factor 7 deficiency. No, that's the other one. That's where the PT would be up, but the APTT would be down. Same with vitamin K deficiency. That would be both. So he is, by default, von Willebrand's. Okay? He is von Willebrand's. So here we go. There's von Willebrand's disease. So you can work it out. And you don't need to know as much as you think you need to know to get it right. But many of you would have looked at that earlier today and gone, I can't do it, it's too much. Where's the answer? The answer's got to be heparin. Where's heparin? I can't see it. Okay, you know, that's what you're looking for, isn't it? You're going down here going, I need heparin on this list. I can't do it. But you can do it. Just be calm and just think through it. You'll be fine. So, coagulations, my last slide. Know your pathways, your extrinsic, your intrinsic, and your common pathway, and which bits, and break down the problem. The whole purpose of TAT is it's aiming to test your understanding. And coagulation, you can see straight away how easy it is for me to write some coagulation questions, isn't it? Yeah? And if they're easy to write, how likely are they therefore to be in the exam? Because I can write some, because some questions are really hard to write questions for, and therefore they don't exist in the exam. But some questions are easy, and this is one that's an easy area, I think. That's it, that's laboratory methods. There's more of me later.